motor neurons are carrying electrical signals away from the brain to the spinal cord. So these are considered to be efferent. We have just two different types. Okay, they're both excuse me. They're both general. So going to skeletal muscle, that is the body wall or soma. Skeletal muscle. So we'll be looking at these in our last unit when we talk about the nerves that innervate breathing muscles and muscles that we use to stand up, muscles for facial expression. And then general somatic, general visceral efferent. And this is going to smooth muscle. Smooth muscle is found in the iris of your eye, in blood vessels, in tubular organs like the intestines and the stomach. Cardiac muscle, obviously, is the heart. And then glands. So autonomic control. Now, as we'll see in our next unit, there are two types of general somatic. They release different neurotransmitters, sympathetic and parasympathetic. Okay, these are considered, I'll just kind of briefly, we'll spend significant time on them, but there is a concept known as dual innervation. And like I said, we'll get back to this. Most organs are innervated by both sympathetic and parasympathetic, and they have opposite functions. So sympathetic increases heart rate. Parasympathetic decreases heart rate. Sympathetic decreases your digestive activity. Parasympathetic increases it. So essentially, sympathetic is active during stress, and parasympathetic brings it back to normal body functions. So another term for sympathetic effects is fight or flight. So your body gets ready to run away. Blood goes to your muscles, your eyes dilate, your breathing and heart rate quicken. Parasympathetic is normal activity, so rest and digest. Okay. However, in the reproductive system, they both work together, so they don't have opposite effects. They're both needed for reproductive. And then sometimes only one of them does both sides. So for blood pressure, you have high blood pressure, your blood vessels dilate by having less sympathetic activity. If blood pressure drops, then the blood vessels constrict by having more sympathetic activity. Okay? So we have mostly, it's a dual innervation that's opposite control. Sometimes they work together, and sometimes only one or the other does the activity. So like I said, in our next unit, we have a significant amount of time for autonomic nervous system. Okay, so that's the overview. Any questions on how this is laid out? So quiz and exam type questions are to be able to assign a particular structure or type of neuron to a central nervous system or peripheral nervous system. And you'll see that as we go through. There's no point listing it all now because that's what our lecture topics are about. As I said, when we get to the cranial nerves, be able to identify what types are functional. So like the trigeminal nerve I talked about is going to be general somatic afferent because it's sensory from the face. And the third branch is also motor to chewing muscles. So that would be general somatic efferent. Okay. The facial nerve has taste fibers, so we would have general visceral, special visceral afferent for taste. It activates the tear glands, so we would have general visceral efferent because it's autonomic. and it does muscles of facial expression. So it'd also be general somatic defect. Right? So we'll use these specific terminologies when we get to the cranial nerves. All right. Now we're going to look at the tissue of the nervous system. And obviously the functional cell of the nervous system is the neuron. But we have specialized connective tissues as well. And so this view that's on the board is taken from the spinal cord. There's a central channel running down the middle of the spinal cord, about the diameter, slightly smaller, of one of these pointer sticks. And then around that we have neurons. So this is a neuronal cell body here. And they have long processes. And often those long processes are covered with a lipid layer. You've probably heard of myelination, myelinated nerves. And that lipid layer gives them a white appearance. So where we have just cell bodies, which are never covered with this lipid layer, we have gray matter. And where we have the myelinated fibers, we have white matter. All right, so you've probably heard about use your gray matter, talking about the brain and neurons. So a cross-section of the spinal cord, just to see where this tissue, this diagram is taken from. You don't have to copy this. This is just to illustrate. We'll be talking about the spinal cord later. So this H shape that I'm drawing is gray matter. Okay? That's where the cell bodies are located in the spinal cord. And right in the center here is the central canal. So this diagram is taken, say, from a region that looks like this. This would be gray matter, this would be white matter, and there's the central canal. Okay? So there are 5 types of cells that are supportive cells. 4 of them are found in the brain and spinal cord, or the central nervous system. 1 is found in the peripheral nervous system. Right? So on page 8, those 4 cells that are listed there are found in the central nervous system. And the picture of the first one is covering up some of the words. So what it says is physical support of the very top picture for astrocytes. It says physical support of neurons in spatial relationships, blood-brain barrier. You can probably figure that out. And then takes up. Takes is the word that is partially covered up there. Takes up. And then the next word is material for synthesis. And then takes up again. And then takes up again for glucose. And then finally the last word is modulate neuronal impulses. Okay, that's kind of what was left behind there. So let's take a look at these areas. Can I erase this diagram here? Almost done? Okay. So the first cell that's listed up there is this one right here. Okay, it's an astrocyte. It doesn't move around a lot. And its key characteristic, it's not so much that it has lots of branches, because all of these do except for these. So it doesn't, that's not unique. But it's unique because of its location between the capillaries inside the central nervous tissue and the neuron. Right? So here would be a capillary. And when we talk about blood vessels, capillaries in the brain, we use the term the blood-brain barrier. And I talked about that briefly when we were talking about junctions. And I said capillaries in the brain have lots and lots of tight junctions, and that creates a blood-brain barrier. So tight junctions were the snap-like junctions, and a loose tight junction was when we had just a few scattered apart, and lots of larger molecules could make their way between them. And tight junctions meant that it had lots and lots and lots and lots of those little snaps. So you had to have a small molecular weight to make its way between those tight junctions. So capillaries in the brain tight junctions. It makes it difficult to treat infections. So I told you my daughter's horse has a protozoan infection. It's called horse. I don't usually use the word equine. Horse protozoan myeloencephalitis. And so this protozoa, which is kind of a cross between bacteria and viruses, somewhere in between, not a cross, but between, its characteristics lie between those two, is eaten by the horse. And from its digestive tract, it migrates up spinal nerves into the spinal cord and takes up residence there and causes an infection of the spinal cord. And so I was asking the vet about, started this medication yesterday, and I said, so how does it get into the spinal cord if you've got the blood-brain barrier? And she said, well, it's a pesticide, and which means, she didn't tell me that, but she told me it was a pesticide, but the pesticides are typically organophosphates, and they're soluble in the lipid material of our plasma membranes, and so they can just move out of the capillary that way. But many antibiotics, proteins, and other structures can't pass across that plasma membrane, and so it's difficult to treat many infections. So astrocytes, notice these processes of the astrocyte, it wrap around and form another layer that enhances the blood-brain barrier. So the astrocyte is multilingual. It has multiple processes, and those attach to the capillary, and then additional processes attach to the neuron itself. So it has this star-like appearance, hence its name, astrocyte. Okay, so it functions in the blood-brain barrier. And the environment of the neuron. So potassium and glucose regulation. Those other functions that I listed on page 8. Any excess neurotransmitters, which are the chemicals released by a cell when it signals. If they remain in the solution, they can cause neurons to fire when they weren't supposed to. So they have to be removed so that when the neuron is exposed to them again, it will fire. And you don't have this, fuzziness in the signal. All right, then we have smaller cells that also have multiple processes. They always remind me of bristle stars, the spiny sea stars. And these are more mobile. These can move around, they're tiny, and they have additional processes. And these are the equivalent, if you will, of the macrophages in the central nervous system. They're phagocytic. So these are called microglial cells. Or if you wish, microglial. So they're going to remove cell debris, not dead cells, proteins that need to be additionally removed as well.
They're not attached.
No, they're going to migrate around. And then that central canal but we also, as we'll see at the end of lecture today, is continuous into the brain, and we have fluid-filled spaces in the brain known as ventricles. So lining the central canal and lining these ventricles of the brain, we have ependymal cells. And ependymal cells have long processes, but they have these kind of pyramidal shapes. And they're found always on the surface of the central canal or the ventricles. And tomorrow we'll talk about certain regions of these are associated with blood vessels and actually make the cerebral spinal fluid. All right? So they secrete and modify the cerebral spinal fluid. So most of the nuclei we see when we look at a histology slide of nerve tissue are going to be these neuroglial cells. Right? The neurons are quite distinctive because they have these large cell bodies, and the nuclei of a neuronal cell body reminds me of an owl's eye. A large nucleus with a very large nucleolus. And we usually don't see the entire neuron, but we'll see parts of it.